Okay, welcome back. So before lunch, before break, we have done 50 questions from our paper, the subject wise test paper. We're moving on to question number 51. We are moving on to question number 51. Okay, so just in the previous slide we had talked about the various lipoproteins and what type of transport they are doing. We had just mentioned the VLDL will carry out the VLDL will carry out the endogenous transport, the chylomicron will carry out the exogenous transport, the reverse cholesterol transport is carried out by HDL. The reverse cholesterol transport is carried out by HDL, whereas the LDL will transport cholesterol to peripheral tissue. It will transport cholesterol to peripheral tissues. Okay, so keep this in mind, the various lipoproteins and the mechanism by which they are transporting. Also remember, VLDL has the marker B100, LDL will also have the marker B100, it will also have the marker B100. The HDL will have the marker ApoA1, the chylomicron will have the marker B48. Important about LDL is you have a single copy of B100 on one LDL particle, single copy of B100 on one LDL particle, whereas in this case you can have multiple copies. In this case you can have multiple copies, whereas LDL will have a single B100 copy on the surface. Question number 52. Vitamin essential for transamination. Vitamin essential for transamination is pyridoxine in the form of pyridoxal phosphate, which is vitamin B6. Pyridoxal phosphate is required for transamination. It is required for deamination. It is also required for decarboxylation. It is also required for decarboxylation, and in a lot of reactions the pyridoxal phosphate also acts as the phosphate donor. In a lot of reactions it acts as the phosphate donor. So if you look at specific reactions where pyridoxal phosphate is acting, it is required for synthesis of catecholamines. Catecholamines means the dopamine, norepinephrine, epinephrine, along with synthesis of GABA and the synthesis of serotonin. All of these reactions will require pyridoxal phosphate. It is required by the enzyme serine racemase. So for the metabolism of serine it is required. It is required for the metabolism of sulfur containing enzyme, methionine, cysteine, both of them require pyridoxal phosphate. It is also required for heme synthesis. It is required for glycogenolysis. It is required for sphingolipid synthesis also. It is required for sphingolipid synthesis also. These are some of the specific reactions where we make use of pyridoxal phosphate. B1 is thiamine, active in the form of thiamine pyrophosphate. B2 is riboflavin, active in the form of FAD and FMN. B12 is cyanocobalamin. Okay, so this is about vitamin B6. Important about vitamin B6. Pellagra symptoms are aggravated if diet contains, see, all of these should have a space in between, which I have highlighted. This part should have a space. Okay, so high amount of leucine will precipitate pellagra. High amount of leucine will precipitate pellagra. Why? Because leucine will interfere with conversion of tryptophan to niacin. This is inhibited by leucine. Leucine inhibits the conversion of tryptophan to niacin, and that is why pellagra symptoms will be aggravated if diet contains high amount of leucine. Most of the leucine comes from dietary tryptophan. The tryptophan which is there in our diet is then converted to niacin. If leucine is there in high amounts, this niacin concentration will fall and you will have pellagra. Okay, Menkes kinky hair syndrome is one of the inherited collagen disorders. It is one of the inherited collagen disorders, and the enzyme lysyl oxidase is not working because it requires copper, and this copper is transported by ATP A7. It is transported by ATP A7 or ATP 7A. ATP 7A is the defective transporter in case of Menkes kinky hair syndrome. In Menkes kinky hair syndrome, the defective transporter is the ATP 7A. The other important inherited collagen disorders include the osteogenesis imperfecta, the Ehlers-Danlos syndrome, the Alport syndrome, and epidermolysis bullosa. These are some other important inherited collagen disorders. These are some of the other important inherited collagen disorders, names of which you must know. Okay. A similar copper binding ATPase defect is seen in Wilson's, where you have the ATP 6A defect. Okay. Tangiers. Now, in most of the cases, you are having in hyperlipoproteinemias increased LDL or VLDL or the chylomicrons, but in Tangiers there is low or absent HDL. In this case is low or absent HDL, and you recall that HDL is anti-atherogenic, so propensity of developing atheromas is quite high. All the following are true about NADPH except. Now NADPH is produced from H HMP shunt and utilized only in the cytoplasm. Because it is utilized only in the cytoplasm, it does not enter the mitochondria, it cannot produce ATP in RBC. So this statement is false. As per the question, your answer would be this. The NADPH stabilizes the RBC plasma membrane by acting as an antioxidant. By acting as an antioxidant, it stabilizes the RBC plasma membrane. The glucose 6-phosphate dehydrogenase deficiency causes decreased NADPH production. This is the first enzyme of HMP shunt. So obviously, if HMP shunt is inhibited, then NADPH synthesis will not occur, because the glucose 6-phosphate dehydrogenase is the first HMP shunt and is required for reductive biosynthesis. Wherever reductive biosynthesis is going on, NADPH will be required. For example, in testes, in the thyroid hormone, in the adrenal cortex, in the adipocytes, in the liver, wherever biosynthetic activity is going on, you will require the NADPH. Wherever biosynthetic activity is going on, you will require the NADPH. In all these places, you are going to need NADPH. Okay, question number 57. Which of the following techniques is used for detection of variations in DNA sequence and gene expression? See, two things: difference in DNA sequence as well as difference in gene expression. Now, the southern blot can identify differences in DNA, only DNA. Northern blot, only RNA. Western blot, differences in protein only. I am sure you know this. Southern is for DNA, northern is for RNA, western is for protein. This is nil. Southern is for DNA, northern is for RNA, western is for protein. So you have snowdrop. The mnemonic that we use for the blotting techniques is the snowdrop. What exactly is microarray? You have a glass slide which is further subdivided into multiple compartments. It is further subdivided into multiple compartments. In this case, we have 64 compartments. Each compartment will have the oligonucleotide, the primers for a specific gene or a specific RNA. So simultaneously you can test for differences in 64 genes or differences in expression for 64 genes. So that's the microarray, and microarray is a technique used for detecting variation in DNA sequence and gene expression. You can do both. You can do both using the same technique. Okay, again it shows the titration curve of the amino acid. We had talked about this previously also, that this shows the titration curve of an amino acid, maybe glycine, it could be glycine, we're not sure. Okay, so as we can see, every time you have the titration, we have the titration. See what happened if you compare this graph with the previous one. What you see is the axes have been changed. In the previous question, the equivalent of OH was on the x-axis, whereas pH was on the y-axis. But here we will just change the orientation. So what you can see here, the compound has two ionizable function. It has two ionizable function. The titration part are the steady state conditions, when there is change in pH, that would be the titration part. So it has 2 ionizable functions, this is wrong. 3 ionizable side chain, again it is wrong. And we had said previously also the maximum buffering capacity of the compound is represented by point A and B. Basically this is a repeat, only thing they have inverted the diagram, so nothing much to discuss here. Moving on to question number 59. Now, deficiency of purine nucleoside phosphorylase causes a syndrome known as the Nezeloff syndrome. Okay, so essentially here you have the cellular immunodeficiency, the T cell deficiency will be there. The Nezeloff syndrome shows cellular immunodeficiency, the T cell development is not adequate. The combined immunodeficiency is seen in the deficiency of adenosine deaminase, the enzyme just before the nucleoside phosphorylase in the degradation of purines. So the adenosine deaminase will give rise to SCID. Deficiency of ADA gives rise to SCID. In the de novo pathway, the first purine nucleotide to be synthesized is IMP. IMP is then converted via 2 reactions into AMP and GMP. Okay, so the first purine nucleotide to be synthesized would be inosine monophosphate, which is then converted to AMP and GMP. UMP is a pyrimidine. It is not a purine. UMP is a pyrimidine. It is not a purine. Question 61. Inhibition of the synthesis of ATP during oxidative phosphorylation by oligomycin is thought to be due to which of the following events? Now, oligomycin is inhibitor of the F0 subunit of ATP synthase. Okay, so it does not block the gradient between NADH-Q reductase and QH2. It does not block the proton gradient. It does not dissociate cytochrome C. What it inhibits is the mitochondrial ATPase, also known as complex 5, which is made up of 2 subunits, F0 and F1. The target is F0 subunit. The target is the F0 subunit. Please note, another important inhibitor of F0 subunit is venturecidin, also known as abomycin. Likely to be asked in your exam, the venturecidin or abomycin is also an inhibitor of the F0 subunit in addition to oligomycin. Venturecidin or the abomycin is inhibitor of F0 subunit. within the inner matrix of mitochondrion. The enzymes of tricarboxylic acid cycle are there except succinate dehydrogenase. Components of electron transport chain are embedded in inner mitochondrial membrane, same with succinate dehydrogenase. Glycogen molecules present in the cytoplasm. Enzymes of gluconeogenesis mostly in cytoplasm, first enzyme in mitochondria, but otherwise mostly it is in the cytoplasm. So the inner matrix of mitochondrion contains the enzymes for most of the metabolic pathways in the mitochondria, including the Krebs cycle, the beta oxidation, the xenobiotic metabolism, the ketogenesis, the bile acid synthesis, minor pathway. So all of them would be occurring inside the matrix, whereas the electron transport chain are embedded in the inner mitochondrial membrane. The different proteins, about 67 different proteins are there, all of them are embedded in the inner mitochondrial membrane with two exceptions, Q and cytochrome C. These are mobile, these are in the free form, they are not embedded, these are in the free form. In hereditary fructose intolerance, there is defect in the fructokinase is associated with essential fructosuria, the phosphofructokinase we have already said, is associated with Thyroid disease. The fructose 2,6 bisphosphatase has a picture similar to HFI, but in HFI the aldolase B is deficient. Fructose 2,6 bisphosphatase have pictures similar to HFI but not exactly the HFI. The HFI is classically associated with aldolase B. The aldolase B is found only in the hepatocytes. And deficiency of aldolase B causes sequestration of the phosphate. Deficiency causes two things: sequestration of phosphates and hyperuricemia. Okay. So which of the following types of mutation generally leads to a truncated protein? Now theoretically speaking all of them will give rise to truncated protein. So what you have to tell us now, most commonly what is the cause for a truncated protein? Which mutation most commonly causes truncated protein? And the answer would be frameshift mutation. See, deletion and insertion, both of them are frameshift mutation. So a better answer is frameshift mutation. Whether it is deletion or insertion, both of them can give rise to truncated protein via frameshift mutation. Missense mutation can cause a truncated protein, but low probability. Okay. So theoretically all of them, the best answer is frame shift mutation. If you go for a PGI exam, then you are going to mark all the 4 options. All of them can give rise to truncated protein. All of them can give rise to truncated protein. Now when you talk about the state of respiratory control, then under different conditions different metabolites are limiting for respiration. Different metabolites and different situations are limiting. Based on that, we have 5 types. We have 5 types of respiratory control, which is given in your handout, and when you check it out, then you will find that at the resting state most of the cells in the body are at state 4. In state 4, availability of ADP is rate limiting in nature. Reason being, what is happening? The ATP is not being utilized at a very fast rate. We have a limited pool of ATP and ADP, so the ADP is not available to carry out the respiration, so the respiration will slow down. When you start using that ATP, more ADP becomes available and the respiration can start again. So at the resting state most of the cells are having limited ADP and therefore they are in state 4. Most of them are in state 4. Okay, question number 66. True among the following is. Now see, this is basically an information dump question. We want to tell you some information, and that is why we have given this question. Hyperamonic condition is considered essential for the severity of hepatic encephalopathy. We told you, as long as ammonia is not there, encephalopathy will not occur. So hyperammonemia is required for encephalopathy. Even if there is hepatic damage and hyperammonemia is not there, encephalopathy is unlikely. So what happens in the brain? The ammonia will get converted to glutamate, and glutamate is then converted to glutamine by the enzyme glutamine synthetase. It is then converted to the enzyme to the molecule glutamine by glutamine synthetase. It is not just the formation of glutamate where the process stops, it is further converted to glutamine. Okay. So in addition there is a trapping of ammonia in alanine. In addition there is trapping of ammonia in the form of alanine. The basic idea is the body wants to diminish the amount of ammonia so that less and less of it is available to deplete the alpha. Less of it is available to deplete the alpha ketoglutarate. Less of it is available to deplete the alpha ketoglutarate. So the answer is all of the above statements are correct. All of the above statements are correct. '67. The gas between segments of DNA on the lagging strand produced by restricting enzymes are rejoined or sealed by. The answer in this case would be DNA ligase. The DNA helicase causes unwinding. DNA topoisomerase removes torsional strain. The DNA topoisomerase removes torsional strain. DNA phosphorylase is not an enzyme of DNA synthesis, rather it is an enzyme of DNA degradation. The other enzymes we have added in your handout. We have talked about the primase, we have talked about the polymerase. So we have added the primase and polymerase in your handout. You can check them out there. Question number 68. Now these are crystals of cystine. These are crystals of cystine. So again, when you check the handout, you will find we have given the crystals for each of them. What does the calcium oxalate look like? The hexagonal envelope shaped crystals are there for calcium oxalate, calcium phosphate, the cystine and triple phosphate. All of them are there in your handout. What are the substrates for PCR? This question is asked repeatedly again and again and again. So you require the deoxyribonucleotides, you require the Taq polymerase, you require the template DNA, you require primers. These primers are DNA in nature. You require coenzyme magnesium, and you require buffer to provide the optimum pH. So these are the substrates required for PCR, and let's talk about the steps of PCR. What are the steps which you require to perform the PCR? These include initiation, elongation and termination, and this cycle is repeated. I would say this cycle is repeated 30 to 40 times. This cycle is repeated 30 to 40 times. The dideoxyribonucleotides are used for gene sequencing. The dideoxyribonucleotides are used for gene sequencing. Now, what Pompey's I previously highlighted, the Pompey's disease is due to acid maltase, or the lysosomal alpha 1,4 and alpha 1,6 glucosidase, together known as the acid maltase. Debranching enzyme we have already seen associated with Cory's muscle phosphorylase, with McArdle's, the branching enzyme with Anderson's. Cory's is type 3, McArdle is type 5, Anderson is type 4, whereas the Pompey's is type 2. So we are emphasizing again and again the glycogen storage disorder, very frequently asked question. Most major metabolic pathways are considered to be either mainly anabolic or catabolic. Which of the following pathways is most correctly considered to be amphibolic? Lipolysis, catabolic; glycolysis, catabolic; beta oxidation of fatty acids, catabolic; citric acid cycle is amphibolic. You can go to amino acid synthesis, or it can go to acetyl coenzyme A. Intermediates can be diverted towards gluconeogenesis also. Intermediates can be diverted towards gluconeogenesis also. The acetyl coenzyme A can either be catabolic or it can go to cytoplasm as citrate, where it participates in lipogenesis. You see, the Krebs cycle is amphibolic. It can carry out breakdown of the acetyl coenzyme A, it can carry out amino acid synthesis, it can carry out lipogenesis, it also participates in gluconeogenesis. So multiple anabolic as well as catalytic pathways operate via the Krebs cycle or the citric acid cycle. Which receptors are present in liver for uptake of LDL? Now this question is not very correctly framed. Which receptors are present in the liver for uptake of, the statement should be lipoproteins, or rather you can say VLDL remnants. Even then the question is not correct, because the ApoE and ApoA are present on the lipoproteins, not on the receptors. And the receptor are identifying the ApoE and ApoA. The LDL receptor, the LRP identify the ApoE and ApoA. So I would say the question is not correctly framed. Which receptor is present in liver should be converted to receptor present in liver for uptake of LDL identifies. Now the question is more correct. Receptor present in liver for uptake of LDL identifies, and then you have the answer as ApoE and ApoA. The LDL receptor will identify both ApoE and ApoA. So we will cut out this question and you read the question like this: the receptor present in liver for uptake of LDL identifies the ApoE ApoE and ApoB100. ApoB and ApoE and ApoB100. The receptor present in liver for uptake of LDL identifies the ApoE and ApoB100. So the correct question is receptor present in liver for uptake of LDL, that is the LDL receptor. This receptor will identify both ApoE and ApoB100. This receptor will identify both ApoE and ApoB100. Insulin promotes lipogenesis. Insulin promotes lipogenesis by increasing glucose uptake in the cell, by inhibiting pyruvate dehydrogenase, by increasing acetyl coenzyme A. Decreasing cyclic AMP is not the direct mechanism. The direct mechanism so insulin promotes lipogenesis by increasing the glucose uptake in G cells, by inhibiting pyruvate dehydrogenase, and by increasing the acetyl coenzyme A. So in this way. Thank you very much. Thank you very much. The increased acetyl coenzyme A will combine with oxaloacetate to give you the citrate, and the citrate will then go from mitochondria to the cytoplasm, where it will split into acetyl coenzyme A, which can participate in the lipogenesis. The cyclic MP regulates glycogen metabolism. It regulates the glycogen metabolism, but it has little role in the lipogenetic pathway. Which reaction in the illustration below occurs in both muscle and liver but has substantially different qualities in the two? Now what happens in muscle and liver is the presence of enzymes catalyzing these reactions. The enzymes which are available to catalyze the reactions in first of all, reaction A, that is conversion of glucose 6 phosphate to glucose, does not occur in both liver and muscle, only in liver, or at most in kidney, not in muscle. So the reaction A is not occurring in both liver and muscle, that is out. Reaction B is occurring in both liver and muscle, but there are two enzymes which are participating here, hexokinase and glucokinase. The glucokinase is present only in two places, liver, beta cells of pancreas. So in these two tissues, conversion of glucose to glucose 6 phosphate can be carried out by both hexokinase and glucokinase, whereas hexokinase is the only enzyme in muscle. So reaction B occurs in both the liver and in the muscle, but different qualities because of the presence of glucokinase. Glucokinase has a high Km and it does not undergo inhibition, unlike hexokinase. Hexokinase has low Km and it undergoes feedback inhibition. By the glucose 6 phosphate. Glucose 6 phosphate will carry out feedback inhibition of the hexokinase. So in this way, reaction 2 or the reaction B is different in the muscle and the liver. Reactions C and D are the same. So your answer will be reaction B. In a critically ill patient, amino acid supplementation will maintain positive nitrogen balance by. Okay, when you start giving the amino acid in a critically ill patient, then this amino acid pool can be used for gluconeogenesis, and this amino acid can directly be metabolized. Also, it can enter the Krebs cycle. But that is not the role of maintaining positive nitrogen balance. What it does is, when amino acid is available, it decreases the protein degradation. Normally when the patient is ill and there is decreased amount of amino acid in the body, the amino acid are obtained from protein degradation. So the first thing the amino acid will do, it will decrease the protein degradation. It will carry out decrease in the protein degradation. Now because lot of more amino acid is available, it will also cause increase in protein synthesis. So amino acid supplementation has a dual action. On one hand it decreases the protein degradation, on the other hand it simultaneously increases the protein synthesis. Okay, so both B and C, increased protein synthesis as well as decreased protein degradation, will help to maintain positive nitrogen balance. They will help to maintain the positive nitrogen balance. This protein synthesis will start incorporating the amino acids, and the decrease in degradation will ensure less nitrogen is getting lost. Now in multiple sclerosis, it is not just the phospholipids and ceramide, but it is phospholipids as well as the sphingolipids. In the demyelinating disease, multiple sclerosis, phospholipids and sphingolipids both are lost. The phospholipids and sphingolipids both are getting lost. It is not just the phospholipid and ceramide. What is ceramide? Ceramide is just the building block, that is sphingosine plus fatty acid. Ganglioside is ceramide plus oligosaccharide plus sialic acid, whereas sphingolipid includes the glycolipids as well as the sphingophospholipids. So all sorts of lipids are getting lost in the multiple sclerosis. Okay, let's which of the following statements concerning glycolysis is correct? Let's read each statement and simultaneously we start saying which is right, which is wrong. The conversion of glucose to lactate requires presence of oxygen. This is wrong. The conversion to lactate occurs in anaerobic condition. Hexokinase is important in hepatic glucose metabolism only in absorptive period following consumption of carbohydrate containing meal. This is also wrong. This is the enzyme glucokinase. When the concentration of glucose is high, hexokinase cannot work. It is the enzyme glucokinase which will work in the post absorptive period. Fructose 2,6-bisphosphate is a potent inhibitor of phosphofructokinase. This is wrong. This is an activator. The fructose 2,6-bisphosphate is the most strong activator of the PFK1. It is the most strong activator of PFK1. The regulated reactions are also the irreversible reactions. These include hexokinase, PFK1 and pyruvate kinase. These are the three reactions which are regulated as well as irreversible. Regulated as well as irreversible. Question number 78. Biological role of metallothionein is to sequester harmful metal ions. Metallothionein is found in cytoplasm of hepatocytes, intestinal cells and renal cells. What it does, it will sequester the cadmium, it will sequester the copper, and it will sequester the zinc. It will sequester the copper, zinc, cadmium. They can even sequester mercury. So these are some of the ions which are sequestered by the metallothionein. Okay, the aluminum, mercury, ammonium, lead. They should be lead, arsenite, phosphate. They are not inhibited. Okay. Question number 79. What is the cycle shown below called? So what is happening? Glucose is going to muscle. In the muscle, the glucose is converted to lactate. Lactate goes to the liver, where it gets converted to glucose via gluconeogenesis, and this glucose can again be used by the other tissues including muscle for meeting their metabolic requirement. This is called the Cori cycle. Eminent male of Parnas pathway is the glycolysis. The pentose phosphate pathway is the HMP shunt. So we are dealing with the Cori cycle. Question number 80, which is not a method of gene therapy. We have just seen fish is a detection tool. We just talked about fish. Fish is a cytogenetic detection tool. It is used to identify segments of DNA, whether they are specific or non-specific segments of DNA. They can be identified by fish, whereas transfection, electroporation, bacteriophage, all of these are tools for gene therapy. Here you can also add the gene gun. You can also add gene gun, which you can use directly for introducing the gene segment inside the cell. You can use it directly for introducing the gene segment inside the cell. Question number 81. Which of the following is not true about cystinosis? In cystinosis, you have the white blonde hair, photophobia is there, Fanconi syndrome is there, the corneal crystals are there, but cystine stones in urine are not common. The cystine is lost in the urine, but cystine stones are not common. Cystine stones are not common. Okay, so this statement is not correct. Cystine can be lost in the urine, but cystine stones are not common. Okay. Cystinosis is different from the classical cystinuria. The classical cystinuria, where cystine stones are formed, is a different condition, where we had said the cola is there, which is in classical cystinuria. This is cystinosis. These are two different conditions. Okay. Question number 82. A patient reports a change in color of urine on air exposure. All are true about the condition shown below. See what is happening on exposure to air. On exposure to air, the blackening of urine is seen. Blackening of urine is seen. So blackening of urine will be accelerated on exposure to selenite. Alkapton bodies are deposited in intervertebral disc, giving rise to fibrosis. Later on, these patients will present with back pain and back stiffness. The urine Benedict test is negative is not true. Urine Benedict test will be positive because it is a reducing substance, and nitisinone is an inhibitor of the enzyme hydroxyphenyl pyruvate dioxygenase. This we had said with tyrosinemia type 1. The nitisinone can also be used for the alkaptonuria. The nitisinone drug that we had mentioned, the anti-BC, can also be used for treatment of alkaptonuria. Although you have to remember that this is a comparatively new drug, the long-term sequelae specifically in children is not yet known. Whether there is any abnormal long-term sequelae or not, this part is not yet understood. The primary role of chaperones. Chaperone belong to a group of proteins called auxiliary proteins. So the question is, primary role of chaperones is to help in protein synthesis, protein degradation, protein denaturation, protein folding. The answer obviously is protein folding. Auxiliary proteins help in protein folding. They assist a newly formed protein for protein folding. Include the chaperones. Chaperones may be chaperonins, or they may be the heat shock proteins. You can have the disulfide isomerase. You can have proline cis trans isomerase. You can even have calnexin. Reticulin, GRP94, and BIP. So these are some of the proteins which assist in folding, and therefore they are known as auxiliary proteins. Some proteins which assist in folding known as auxiliary proteins: the chaperones, the disulfide isomerase, the proline cis trans isomerase, the calnexin, calreticulin, GRP94, the BIP. So these are some of the important auxiliary proteins. Okay, which of the following is insulin dependent for its function? Which of the following is insulin dependent for its function? I had this previously also discussed that the GLUT4 is insulin dependent. Now what do you mean by insulin dependent? See, this is the cell membrane, let's say, and here you have the GLUT4. So as long as insulin is there, the GLUT4 will be there. If insulin is not there, then what will happen? The receptor is internalized. The receptor is internalized. So if insulin is there, the receptor is there on the surface. If insulin is not there, the receptor will go inside in the form of vesicle. So that is what you mean by insulin dependent. It is not the activity which is insulin dependent, but it is the presence on the cell membrane which is insulin dependent. Okay, so GLUT1, 2 and 3 are the other facilitative transporters along with GLUT5, which can participate in the transport of glucose. The GLUT1, 2, 3 and 5 are insulin independent, whereas GLUT4 is insulin dependent. I previously said that the GLUT4 is present at the cardiac and skeletal muscles. It is also present in the adipocytes. So in these tissues, the uptake of will only occur when insulin is there. Insulin is there when there is excess glucose. So these tissues will take up glucose only when excess of glucose is available. Okay, question number 85. Which of the following diagrams depict reciprocal translocation? And we have 4 diagrams, diagram A, B, C and D. So in diagram A, what you see, a segment from chromosome number 2 is transferred to chromosome number 1. The corresponding segment is transferred to chromosome number 2. This is the classical description of translocation. If you look at others, equivalent exchange is not occurring in these 2 chromosomes. Equivalent exchange is not occurring. This is Robertsonian translocation. Where is the second chromosome? This is known as Robertsonian translocation, where unequal exchange is occurring. What is happening here? Most of this chromosome is transferred to the second chromosome. This is called Robertsonian translocation, commonly implicated in Down syndrome. The Robertsonian translocation is commonly implicated in Down's syndrome. Question number 86. Phospholipid associated with mechanism of hormone action is, the answer is phosphatidyl inositol. See what happens. Glycerol associated to fatty acid 1, associated to fatty acid 3, associated to phosphate, which is associated to inositol diphosphate. Now when the this is the phosphatidyl inositol. When cleavage of phosphatidyl inositol occurs, it occurs here, giving rise to IP3 and diacyl glycerol. 2 fatty acids attached to glycerol is the diacyl glycerol, and you know very well that diacyl glycerol and IP3, both of them are second messengers, and therefore they are associated with mechanism of hormone action. Second messengers associated with hormone action. So phospholipid associated mechanism of hormone action is the phosphatidyl inositol. The answer would be phosphatidyl inositol. Question number 87. Which of the following enzymes is used in recombinant DNA research for homopolymer tailing? Now tailing will be done at the end. And the enzyme used is terminal transferase. The enzyme used for tailing is terminal transferase. The reverse transcriptase acts on RNA to generate DNA. Only the terminal transferase will be able to act. The S1 nuclease, the correct term should have been the S1 nuclease. It actually degrades the DNA. And the polynucleotide kinase transfers phosphate from ATP to DNA or RNA. The polynucleotide kinase will transfer the phosphate from ATP to either DNA or the RNA. Okay, so the correct answer is terminal transferase. Now recall previously we talked about technique called microarray, where I told you on a plate you have multiple segments and each of the segment can undergo independent amplification of DNA or RNA, which was the microRNA. Once the amplification is done, then we label them using fluorescent dyes. So this is also a microarray. In fish, cells seen under microscope in ELISA we have a colored plate. In Western blot assay we have a gel labeled with antibodies. We have a gel which is labeled with antibodies. Okay, so in the above diagram what we are seeing is what I had described previously. You are having a plate. This plate has multiple sections. And each section is, you can say, an independent reaction well. So once the reaction is completed, you label them according to the reaction, and this is the result that you get. So we are looking at a microarray. In question number 88, the diagram shows a microarray. The color intensity correlates with the amount of expression, or if there is no expression. Okay, question number 89, which of the molecules is responsible for cell-cell adhesion? Now, among the given options, the answer would be fibronectin. Besides fibronectin, you can have integrins, you can have cadherins, you can even have selectins. Okay, so these are some of the components which are required for cell-cell adhesion. The inactin is found in the extracellular matrix. The laminin means, extracellular matrix means basement membrane. The laminin is also present in the basement membrane, whereas lignin is present in the food. It is a heteropolysaccharide in diet. Question number 90. The following graph represents the effect of substrate concentration on initial velocity of an enzyme catalyzed reaction. Wrong statement about this graph is, first of all, this graph is drawn as per the Michaelis-Menten equation, synthesized as per the Michaelis-Menten equation. So let's read each of the options and then we will describe which is right and which is wrong. The curve is hyperbolic in shape. This is correct. The curve is hyperbolic in shape. You can see the curve is hyperbolic. The question mark here represents Km of the enzyme. The question mark here represents Km of the enzyme. This is also correct. the Km is the substrate concentration at which half the maximum velocity is obtained. So you can see the Km is the substrate concentration at which Vmax by 2 is achieved. So B is also correct. At point C, only a small amount of enzyme is present as the enzyme substrate complex. This is false. At point C, most of the enzymes are present as enzyme substrate complex. That is why the rate of reaction is quite high. The rate of reaction will depend on the formation of enzyme substrate complex. More is the rate of reaction, more will be the number of enzyme substrate complex. So obviously at point C you will have more enzyme substrate complex. This statement is not true. At point C, the velocity depends solely on the rapidity with which product dissociate from the enzyme so that it may combine with more substance. This is also correct. So faster they can dissociate, more the enzyme can participate in the reaction and more will be the formation of product. So for question number 90, the correct answer is C. C is an wrong statement. C is a wrong statement. Question number 91. Lake syndrome is also known as subacute sclerosing or necrotizing encephalopathy. So it is a type of mitochondrial disorder, and in this case what is happening, the glycogen accumulation occurs. The pyridoxine, sphingomyelin lipoprotein is not related to Lake disease. Lake disease is due to the defect in mitochondrial DNA. Question number 92. This is a very frequently asked question related to the quaternary structure of protein. So the questions on quaternary structures of protein are commonly asked. So the arrangement sequence of amino acid in the polypeptide chain is the primary structure. Interrelation between amino acid in a single polypeptide chain would be secondary or tertiary structure. The interrelation of amino acids in two polypeptide chain interrelation of protein with more than two polypeptide chains is the quaternary structure. Interrelation of amino acids in two polypeptide chain. This can also be quaternary structure. But the D is more correct answer. The D is the more correct answer. If given an option, you can go for both C and D, but however D is the more correct answer. D is the more correct answer because here we are not talking about arrangement. So you are not talking about the shape. So until you talk about shape, you are not talking about quaternary structure. The quaternary structure is not just the interrelation of amino acid. It talks about arrangement. It talks about arrangement also. It can be 2 or greater than 2. Here if we had used the term arrangement, then C would also have been correct. Question number 93. A mutation to which of the following sequence in eukaryotic messenger RNA will affect the process by which the 3' end of poly A tail is added to messenger RNA. Now the poly A tail is added to messenger RNA following the sequence of AUAAA, following the sequence of AUAAA, alright, whereas the sequences CAAT, CCA, GGGG, CG, these are specifically found in the promoter region, whereas this sequence is found at the end of messenger RNA, where the tail is attached, meaning your answer should be the AUAAA. Question number 94. In terms of enzyme kinetics, the following plot is of 1 upon initial velocity as y as a function of 1 upon substrate concentration as x. This plot is correctly named as, now this is a derivation of the Michaelis-Menten equation, where you can see the y-intercept, the line crosses the y-axis at a point. You also see the x-intercept and a very specific slope. These are used to describe the Lineweaver-Burk plot, because here we have a reciprocal value on the x-axis. We also have a reciprocal value on the y-axis, meaning both the axes show reciprocal values, known as the double reciprocal plot. So the correct answer should be both A and C. This graph is known as Lineweaver-Burk plot, which is also known as the double reciprocal plot. Alright. Now, rotting fish urine odor. Rotting fish urine odor is one of the important abnormal odors of urine, which is seen in trimethylaminuria. In Hawkinsinuria, you have chlorine like or swimming pool water like odor. In phenylketonuria, you have mousy or musty odor. In MSUD, you have burnt sugar or caramelized sugar odor. The two can be there. Sometimes you can also get asked about isovaleric aciduria, where you have sweaty feet odor. Tyrosinemia we have already said. You get boiled cabbage like odor. In addition, in cystinuria, you can get sulfur odor or rotten egg odor. This is due to hydrogen sulfide. The characteristic odor that you find. Okay, so these are some of the important abnormal urinary odors that you can see in inborn errors. These are the important characteristic odors that you can see. Activity of all of the following enzymes is increased in diabetes state, except, now in diabetes the condition is apparent starvation. So when there is starvation, you will have gluconeogenesis. The pyruvate carboxylase, the PEP carboxykinase and glucose 6 phosphatase, all of them are enzymes of gluconeogenesis, and therefore their activity will be increased. Whereas pyruvate dehydrogenase, it proceeds with glycolysis, takes it to mitochondria and induces the Krebs cycle, requires insulin action. So the answer would be pyruvate dehydrogenase. The activity of pyruvate dehydrogenase will be slightly diminished, whether the other enzymes of gluconeogenesis will be upregulated. The another enzyme, the final enzyme of gluconeogenesis besides pyruvate carboxylase, pyruvate carboxykinase, last enzyme is glucose 6 phosphatase. The third enzyme is fructose 1,6 diphosphatase. So these are all the enzymes of gluconeogenesis. These are all the enzymes of gluconeogenesis: the pyruvate carboxylase, the PEP carboxykinase, the fructose 1,6 diphosphatase and glucose 6 phosphatase. Out of these, only the pyruvate carboxylase is there. Only the pyruvate carboxylase is there in the mitochondria. All others are in the cytoplasm. All others are in the cytoplasm. Presence of which of the following in expression of vector ensures increase in yield of recombinant protein produced? Now if you have an inducible promoter, you can induce the promoter and more of the protein will be synthesized. So this appears to be the correct answer. If you have a gene coding for protease inhibitor, not going to help. Translation initiation signals, if transcription has not occurred, translation initiation signal will not help. Transcription and translation termination signals, again not going to help until unless the transcription has started. Termination itself is not going to help. So the best answer is inducible promoter. The inducible promoter will ensure that there is an increase in the yield of recombinant protein. Which of the following is true? Inhibition of acetylcholine esterase by malathion can be reversed by increasing acetylcholine. This is wrong. This is an irreversible inhibition. The irreversible inhibition is there, so this is wrong. Sulfanilamide reduce folate synthesis irreversibly. This is wrong. This is a reversible inhibition. Ethanol inhibits aldehyde dehydrogenase when used in methanol poisoning. It does not inhibit displaces methanol. Fluorostate competitively inhibits aconitase. This is mostly true. However, the question should say non-competitively. Fluorostate non-competitively inhibits aconitase. The correct answer is fluorostate inhibits aconitase, but the inhibition should be non-competitive. It is not competitive in nature, rather non-competitive. Okay, so in lac-operon, first I am sure you know a little bit about lac-operon. There are various molecules. One of them is the catabolite gene activator protein, and very frequently you are asked which is the positive regulator, which is the negative regulator, which is the promoter, so which is the derepressor, which is the repressor. Like that, a lot of questions are asked. I will quickly go through them and come back to this question again. In the lac-operon model, we have the repressor molecule. First of all, we have the repressor molecule. Repressor molecule is the negative regulator. Repressor molecule is the negative regulator, and it represses the expression of lac-operon. So the first term is negative regulator. The answer is lac-I repressor. Lac-I repressor is the negative regulator. Second term is inducer. Lactose is the inducer, and it de-represses the lac-operon. It removes the repression, so it is called de-repression. Okay. Activate, inactivate at the level of enzyme. Repression, de-repression at the level of genes. At the level of genes, you have the repression and the de-repression. Now, induction by lactose is based on its shape. So if you have molecules similar to lactose in shape, they can also cause induction of the lac-operon model, and these molecules are called gratuitous inducers. Commonly used in genetic engineering in place of lactose. In place of lactose, we use lactose analogs. These are called gratuitous inducers. These are called gratuitous inducers. They have shape similar to lactose, but themselves do not get metabolized. Themselves do not get metabolized during activation. And last is the cap-camp regulator, which is a positive regulator. It is required for optimal gene expression. The cap-camp regulator is required for optimal gene regulation. This is a positive regulator. So please note, negative regulator, positive regulator, and inducer. We have three things: the negative regulator, the positive regulator, and inducer. Let's go back to the question. So the lac-operon is the positive regulator. It is responsible for positive regulation. About lac-operon, you should know that lac-operon is activated when two conditions are met. Number one, glucose is absent, and number two, lactose is present. So lac-operon will be activated when glucose is absent and lactose is present. When glucose is exhausted, there is a lag phase. After that, lactose metabolism starts. Lag phase represents time required for cyclic AMP synthesis, transcription, followed by translation of lactose metabolism enzymes. So the graph is something like this. Population versus time. So when glucose is there, the population is increasing. When glucose is exhausted, there is a lag phase, and then the population starts increasing again. So this is glucose, this is lag phase, and this is the metabolism of lactose. So you get a lag phase. The lag phase is the time required for cyclic AMP synthesis, the transcription followed by translation of lactose metabolic enzymes. So what do you see in the following fish technique? What do you see in the following fish technique? So what is happening? On one of the chromosomes, both the arms having similar shape, the copies here having similar shape. The second chromosome, you can see the green dots. On both the arms you can see green dots, both the arms show red dots. but then you see one chromosome having both green and red dots. So both green and red dots will come due to translocation only. We had seen there is exchange, mutual exchange of genetic material. So what is happening? One green dot has been given here, and in return one red dot has been taken. So when you see the chromosome now, one of the dots would be red, the other dot would be green. This is what we are seeing here. So this is translocation. So you are seeing translocation in this following fish technique. Please note, I told you the fish technique is a cytogenetic technique. So we are not destroying the cell, we look at the chromosomes inside the cell. Typically we are using the metaphase. If you want to study the chromosomes, we use the metaphase. Otherwise any other phase can be used. If you are not doing the karyotype, if you are not looking for a specific modification in the chromosomal structure, then you can use any of the phases. But if you want to analyze the chromosome, then you must use the metaphase, because in the metaphase the chromosomes are most distinct, their shapes can be easily recognized. You can identify which chromosome are you dealing with, whether it is chromosome 1, 2, X, Y, whichever chromosome is there, that can easily be identified in the metaphase. So the best time to do the fish is in the metaphase. You can look for specific genes, you can look for specific defects. If you are not specific about the chromosome, you just want to identify the presence or absence of a gene, you don't want to look whether it is present on the correct chromosome or not, then any phase. In that case you can use any phase when you don't need the chromosome identity. Then you can use any phase if chromosome identity is not required, because fish will find out its target, it will label the correct target, and you can see the fluorescence. The fluorescence will jump out at you when the correct target is identified. Okay, so that was the last question from biochemistry. After this we are going to start with the discussion of forensic paper. Okay, again I would like to say, if you have any doubts, any confusion, we would prefer you go to the DBMCI Premier page. If you have any doubts and you don't want to put it on the Premier page, then you can send me a personalized message. We prefer it to be having on the Premier page because other students can also participate, but some of you are not very comfortable on the Premier page, so you can send them to me via message, or you can even mail them to me. So take care and best of luck. Sure.